Hello everybody and welcome to today's webinar, joint hosted by Abberior and Jackson ImmunoResearch. So my name's Evelyn and I'm an application specialist here at Abberior, and I'd like to say thank you very much for joining us to learn more about the benefits of using nanobodies in your super resolution research. So we've got two great talks coming up today from Kristina Kramer and Wolfe, and these have been pre-recorded, but we will be having a live question and answer session at the end of our webinar. So if any questions come up during the talks, please feel free to pop those in the chat box and we'll come to those later on in discussion. So with me now I have Wolfe from Jackson ImmunoResearch, who's calling in from the US. So thank you very much for being here with us, Katie.
Yeah, thanks Evelyn.
And so Jackson has been a really trusted supplier of antibodies for quite some time now, a few decades. And so what do you think makes Jackson special?
Yeah, thanks. That's a great question. So Jackson has been making antibodies pretty much since secondary antibodies have been made. So we've been owned and operated by our original founders for over 40 years. We're really proud of our products. We stand behind them and we have a lot of expertise making them because we've made them for a long time and we've really actually helped develop the technology to make and engineer polyclonal secondary antibodies. So, you know, we're sort of expanding our horizons now. We still have, you know, our solid portfolio of secondary antibodies, and we're now also expanding out into sort of monoclonals, recombinant type of things. So in the future, Jackson will still have our, you know, portfolio of traditional antibodies that we continue to make with quality in mind, with our techniques that we have known and generated, but we'll also be expanding into new areas as well.
So that was really interesting to hear. I guess a lot of your customers are also going to be research-based.
Oh, yeah.
How does that tend to shape the products and the direction that Jackson moves?
Also a really good question. Our technical service is actually made up of not a separate department. It's actually the people that are making the antibodies, so our R&D staff as well as some people from manufacturing. So we talk directly to our customers when they have questions about product selection or something's not working for them, or if they have ideas. So actually a lot of our ideas, well, at least some of them, come directly from our customers and how they're using the antibodies. So we love to get ideas and feedback from our customers. We're constantly asking, you know, answering the questions they're asking, but then we ask them, is there anything else, you know, that would be cool to use in this assay? Maybe it's a certain type of assay we haven't really heard of or done ourselves before. Definitely a lot of our customers, like you said, are research-based, and we get a lot of feedback from them in terms of shaping, you know, the future of what we make.
Oh, great. And I guess just a question about you and how you work at Jackson. What's your favorite thing about being part of the company?
Oh, yes. So it's a super fun place to work. I've been here for a few years now, and I've been in R&D the whole time, which is a really cool department to work in, but I really love how cross-functional it is. So this is a small company, you know, small-ish, but we have lots of different hats. So I work in R&D, but I also work very closely with manufacturing, with quality assurance, with customer service. So it's been interesting to learn a lot about our business. And the best part about working here, to me, is the reputation that we have and being able to stand behind our products and being proud of what we make. So we have a lot of customers in the research area, like we just talked about, and it's really cool to be able to give them tools to do research, just like I've done in the past. But we also have a lot of customers in the diagnostic space, and that business is expanding for us. So it's also really cool to be, you know, make products that you can be proud of that are used in things like COVID tests for lateral flow assays or diagnostic tests for cancer. So both aspects are really cool, but yep.
Yeah, thank you very much for talking with us, Katie, and I'll now let you give your talk and give a bit of an insight into Jackson and nanobodies.
Okay, yeah, thanks, Evelyn.
Thank you.
So thanks everybody for tuning in to our webinar today. Today I want to talk to you a little bit about Affinipure VHH secondary antibodies, which is a really cool new product that we made here at Jackson ImmunoResearch. So I'll tell you a little bit about what it is, and then talk also a little bit about how to use it. And then my colleague at abberior will tell you a little bit more of how to use it in a very cool application for super-res microscopy. So Affinipure VHH secondaries, they're VHHs. So what's a VHH? Immunoglobulin G, IgG, the general structure of this is the traditional, antibody structure that you're probably familiar with. 2 heavy chains paired with 2 light chains. The heavy chain has 3 constant domains, CH1, CH2, CH3, as well as a variable domain. And so for each of these heavy chains, there's a light chain paired with it. These each have a variable domain and a constant domain. So this is in different host species such as humans, rabbits, mouse, goat, donkey. So your traditional secondary antibodies that you would get from us would be these whole molecule IgGs. In 1993, there was a group in Germany that found out that alpacas, llamas, dromedaries, these are called camelid species, they actually have, as well as the canonical IgGs, some subclasses that are heavy chain only antibodies. So they've lost their CH1 domain, and so they don't need to pair with the light chain. They're stable by themselves without the light chain. So these have just the CH2 and CH3 domains, the 2 constant domains, as well as the variable domain. So that variable domain is VHH, variable heavy only. And since then, that group, as well as others, have gone on to use that very unique property that the VHH itself, which is very tiny, 12 to 15 kilodaltons, can be expressed in a stable and soluble manner just by itself. So it doesn't need a light chain pairing. You can just express it as a single domain. So it's a single domain antibody. You'll hear this as a VHH, a single domain antibody, or a nanobody, which is coined by this group actually that had first discovered this property in camelids. So in this talk, I'm going to display this sort of graphic. So instead of this little oval structure, it's going to look like this triangle structure. And here you'll see 3 little fingers sticking out of one side of the triangle. And that designates the paratope of the VHH. So those 3 little fingers sticking out are actually complementarity determining regions. So there's 3 of these, CDR1, 2, and 3. And a very unique property of these nanobodies or VHHs is that the CDR3 is extended on average larger or longer than traditional antibody CDR3. So what that means is these antibodies, in addition to being very tiny, can kind of reach in places that traditional antibodies may not be able to reach. Additionally, in these slides, you'll see sort of this little star connected to the triangle, and that's just designating the fluorophore or the label that's attached to the VHH itself. So how do you use these VHHs that are labeled? So we generate these as tools for researchers. Many people have expressed these VHHs recombinantly, and they're being used in lots of therapeutic applications, but they also are really useful as a tool for research. So one way you can, in an immunoassay, you could use these is, like other immunoassays, you have direct and indirect labeling. So if you had a VHH primary that was specific for your target of interest, which in this depiction here is the little blue circle, your VHH can bind to the target of interest directly. So the drawback in this situation is that you have to have a VHH to your target of interest. And of course that field is expanding and there's more and more primaries available, but you are limited by having a VHH to your target of interest, as well as having it labeled with the color that you want. So say you do have a VHH to your target of interest, you also have to have it labeled with the color you want. So again, sort of looking at the other side of the coin, this is one of the main reasons, or there's many reasons to use a secondary antibody, but one of the reasons is to get around this limitation of having a primary that's labeled. So say you've got your whole molecule primary, which here is the red antibody. So you've got a primary mouse anti-target 1, mouse anti-target 2, mouse anti-target 3, rabbit anti-target, whatever. So then you can have a secondary that's anti-mouse, a secondary that's anti-rabbit, and your secondaries themselves are labeled, so you can change whichever of those targets to whichever color you want. So it really expands your ability to do a multiple labeling situation. So if we take this one step further and apply the concept of these VHHs as a tool, so what we've made are secondary reagents that are VHHs. They're specific for human, mouse, or rabbit, so you can use these together in a multiple labeling scenario, but they're much smaller. And like I said before, they can kind of reach targets that perhaps a whole molecule might not be able to get to. So you have the benefits of being small, as well as the benefits of having a secondary antibody. Why would you want to use something very small? What are the benefits of using a very small secondary? So in this scenario where you have indirect labeling, in an IHC situation, which is immunohistochemistry, you have a tissue section that's a certain thickness. So say you have a 2 micron section of a tissue, as opposed to having just a monolayer of cells. You actually need to get your antibody into that tissue and also be able to wash it back out. So something that's, this is done very traditionally with whole molecules, primaries and secondaries, very successfully. But a benefit of having something small is you could do this perhaps easier, perhaps less washing. You might not need to use as much antibody. So in this slide here, what I'm showing on the top is the traditional whole molecule primary and secondary. This was done in mouse sections of spleen, and we're looking at a mouse anti-CD8 primary. So the brown sections, the brown staining that you see here is the CD8 signal. So in this top section on the left, you see sort of a zoom out the whole picture of the mouse spleen section, and then on the right is zoomed into these different nodules, so you can see the positive brown for the CD8. Now on the bottom, what we've done is the same scenario where we have mouse anti-CD8, but the secondary in this case is the biotin conjugated VHH anti-mouse. So this is a smaller secondary than the one on the top. And you can see you still get, if you look in the zoom on the right side, very nice strong brown signal here from the chromogen, and just a little bit less browning of the tissue out around it. So if you look in the regions that are not supposed to be brown, they're a little more blue. So again, the top scenario works well, very traditional setup, but in the bottom you see a little bit cleaner or clearer of a signal. So again, this just highlights the fact that these can easily penetrate where they need to be, and also can be easily washed out. So what is another benefit of being very small? So this will be highlighted in the other part of the webinar, but I just wanted to introduce it here. These immune complexes, where you have a primary followed by a VHH secondary, are smaller, so there's smaller linkage error versus having a whole molecule primary and a whole molecule secondary. What does that mean? What's linkage error? That means basically the size of your immune complex and how far your signal is away from your target. So you can see here just sort of by the size of these circles around the little cartoons, this size of immune complex is smaller than this size. And again, you'll hear more about this in the next part of the webinar, but this is very, very, very useful for applications such as super-resolution microscopy. Another unique feature I'd really like to highlight about these particular VHH secondaries that we make at Jackson is that the fact that they're polyclonal. So as I had sort of talked to Evelyn about before, we specialize in polyclonal secondaries. We have a long history of this. We're really good at making them, and so we thought, why don't we make the VHHs polyclonal as well? So the power of the polyclonal is that when you have a scenario where you have your monoclonal VHH secondary, so you have a monoclonal VHH anti-mouse or anti-rabbit, you only have one binding epitope. So if your target, again, is an IgG, you have two options for that VHH to land on. So you can only get a max of two secondaries onto that primary. So if you can imagine the size of a VHH versus a whole molecule secondary, there's a lot less real estate on the surface of that antibody. So that limits the amount of fluorophore or label that you can put onto the antibody. So you can get maybe 1, 2, 3 fluorophores onto a single VHH as opposed to the whole molecule. So say you want to be small, but you also want a bright signal. So here comes the power of the polyclonal, where you have that limitation still of having only a few fluorophores on each VHH, but now you have lots of epitopes to lay your VHH upon. So it really just gives you a brighter signal than the monoclonals. The final feature of these VHH secondaries that I wanted to highlight is the fact that in addition to being polyclonal and very small, we've also cross-adsorbed these, and this is another thing that Jackson is really well known for, and we have done this for a long, long time, where we have lots of different polyclonals available that can be used for multiple labeling. And we solid phase adsorb, so we put them over the antibody that we don't want it to bind to. So say you have an anti-human and you don't want it to bind to mouse and rabbit. So this is this product line has that feature that they're cross-adsorbed, that they specifically label the target and not off target. So in this little cartoon here, you can see the rabbit is supposed to be the purple and the mouse primary is supposed to be the yellow, and you have your anti-mouse and anti-rabbit only landing on their target of interest, and they cannot bind to the opposite one. So in addition, like I said, being small, being polyclonal, you can use them together. So you can have multicolor images that are generated. So I just wanted to show a few examples of that before I let you guys go. So here in this example, we have, again, we're in immunohistochemistry of mouse brain sections this time. So it's 2 micron thick slices of mouse brain. And we've stained these with mouse primary for nu-N and a rabbit primary for tyrosine hydroxylase. And then we've come in with Cy3 VHH anti-mouse and Alexafluor 488 VHH anti-rabbit. And what you can see here in these images over on the left is the single channels, and on the right is the merge with the DAPI staining that's blue. So the Cy3 anti-mouse is red, the Alexafluor 488 anti-rabbit is green. You don't see any overlap, you don't see any crosstalk They stain only where they're supposed to be staining, and they don't stain where they're not supposed to be staining. And you also get a very nice bright signal. So a final example, just another multiple labeling example. This was done by a collaborator at University of Pennsylvania School of Medicine. And this, again, is in IHC, so very useful in penetrating tissue. This is a section of a mouse lung now, and we're looking at mouse anti-smooth muscle actin and rabbit anti-E-cadherin. And the actin is sort of magenta or purple in this image, and the E-cadherin is the green, and you don't see any green and purple overlapping. They're separate and generate beautiful images, but also excellent data to use for whatever your research purpose is. So I just wanted to end with showing the different color options that we have for these. So we have the biotin conjugate, which was what I highlighted in the IHC of the mouse spleen. When we did that experiment, we were using that with an ABC complex and then a DAB chromogen. So that whole experimental setup is totally feasible, and it's being used. And we've done that with a few different types of tissue, in addition to the mouse spleen, and it works great. So in addition to that, we have sort of the spectrum of colors. We've got Dialate 405 down on the UV end. We've got the greens, oranges, and reds that people like to use. And then we've got the near IR dyes as well. In addition to the immunofluorescence, the ICC and IHC that I've mentioned, again, you can use these for super resolution microscopy. You'll be a bit limited here. So some of these fluorophores are okay with super res. Other fluorophores that can be used for super resolution, we're really excited to work with abberior, and they're going to be offering those particularly for that application with different fluorophores. The near IR that I talked about, so the Lexaflor 680 and 790, these can be used for Western blotting, and we've actually done multiple labeling Western blotting as well as in-gel Westerns. So if you think about something small penetrating a matrix, you can use these for in-gel Western blotting if you're doing anything with that. And we've also used these for flow cytometry. That's the end of this portion of the webinar. And then we're going to move on to hear more about some really cool things you can do with super resolution microscopy from Kristina at abberior. So thanks, Evelyn, for letting me discuss our new products.
Thanks very much, Katie. It was really great to hear from you about the products. So with me here I have Kristina Kramer, who is an application specialist here at abberior. Hi, Kristina.
Hi.
And I know that before you started at abberior, you had experience with a range of different microscopy techniques. So how has learning STED microscopy compared with some of those other techniques, and how quickly do you think you picked it up?
Yes, so in the past years I worked a lot with different microscopy techniques like atomic force microscopy, but also confocal laser scanning microscopy. And some of these methods were really easy to learn, while others were more complex or complicated concerning handling and software. So when I started at abberior, I had no practical experience in using STED microscopes, and I was really excited how fast I was able to learn the techniques and also the handling with the software and the instrument, and how fast I was able to take beautiful pictures and also quantitative data.
And so now that you've got more of a handle on STED microscopy, you're doing more measurements regularly, what does kind of a typical day at abberior as an application specialist look like for you?
That's a very good question. So I think there is nothing like a really typical day, because my work, I would say, is very broad and diverse. What I do is a lot of cell culture, but also measuring on our instruments, like for example the Steadicon, but I'm also included in planning exciting novel projects, and what is also a great part is to stay always up to date in the development of our fields. However, what for me starts with in a typical day is drinking a coffee in the morning.
That's great, thank you. So I'll let you get on and give your presentation. The floor is yours.
Thank you. I will talk today about super-resolution microscopy like stacked microscopy and different labeling strategies, including VH8 fragment antibodies, so-called nanobodies from Jackson ImmunoResearch, in combination with our abberior dyes, which are very well suited for multiplexing and also super-resolution microscopy. To begin with, I would like to introduce STAT microscopy, where we aim at the investigation of small cellular components by means of fluorescence. So in conventional light-focusing microscopes, like for example fluorescence microscopes, we are limited due to the diffraction of light to 200 nanometer in resolution. This is due to the fact that light propagates as a wave, and an objective lens is not able to focus light into a point like a single molecule, which would allow for molecular resolution. In reality, the area of a focused laser in diameter equals half of the wavelength of the used light, and that means that all molecules that are in this diffraction area are at the same time excited and emit fluorescence. In STAT microscopy, to investigate even small cellular elements, we aim to reduce this area and only want to allow molecules within the center of this ROI to emit fluorescence. We do this by the application of a second red-shifted laser, which emits photons that have an energy that is not high enough to excite molecules so that they can emit fluorescence, but that fits very well the energy gap between the excited state and the ground state. By adjusting the laser into the famous donut ROI, we have a low intensity of this red-shifted laser in the center, which allows the molecules that are present here to emit fluorescence. In the area of the donut ROI, we have stimulated emission, which means that we only get signal from the molecules in the center of this ROI, and thereby we can obtain high-resolution images by scanning both lasers through the sample, which is below 20 nanometers in resolution. To investigate now cellular structures and components in cells, we have to add organic molecules or organic dyes or fluorescent proteins to our target of interest. This can be done by various different labeling strategies. On the one hand, some of them need previously genetic modification, like for example the click chemistry, where we add unnatural amino acids to our protein sequence of interest, which then can be coupled to organic molecules obtaining or harboring a reactive group. Another option is the expression of fusion proteins of the target protein of interest, for example with fluorescent proteins like GFP, but also with self-labeling proteins or self-labeling enzymes like Halo or SnapTag. These ones are not fluorescent by themselves, but harbor a recognition domain, which can recognize then a fluorescent organic molecule that has a certain reactive group and bind covalently or non-covalently to the fluorophore. Another option are the small molecule probes, which are small drugs, oftentimes peptides, that also are linked to an organic dye, and that these can stain by interaction or intercalation different structures like actin or tubulin, for example. A very commonly used group of labeling strategies is based on antibodies and called immunofluorescence. Here we have on the one hand the direct labeling and the indirect labeling. For the direct labeling, we have one antibody that is coupled to the fluorophores and recognizes the structure or the protein of interest, for example. In indirect labeling, we have a primary antibody that is not labeled with a fluorophore and a secondary antibody that carries the fluorophore and can recognize the primary antibody. In direct labeling, we have a very easy staining protocol. However, sometimes the signal intensity is very low. Here we have an advantage in indirect labeling, since more than one secondary antibody can bind to a primary antibody, which leads to signal amplification. However, in this case, we have a large complex that can lead to a higher linkage error, which we can improve by the use of so-called VHH fragment antibodies or nanobodies. These nanobodies are, on the one hand, 10 times smaller compared to the antibodies, and moreover, the VHH fragment antibodies from Jackson ImmunoResearch are polyclonal, which means that more than one nanobody can bind as well to the primary antibody, which leads to very good signal amplification. In the next slide, I would like to show you a comparison, or an exemplary comparison, of a labeling strategy with the secondary antibodies compared to the secondary nanobodies. So here we stained tubulin filaments with primary mouse antibodies, on the one hand with secondary antibodies in combination with our abberior star red dye, and on the other hand with secondary nanobodies in combination with our abberior star red dye as well. What we found by evaluating the diameter of the tubulin filaments was that we obtained a 10 nanometer smaller diameter for this labeling approach, in contrast to the labeling approach using the secondary antibodies, which shows that we really can reduce the linkage error when we use nanobodies in contrast to the usage of secondary antibodies. Besides single color staining, we also can perform multicolor imaging or multiplexing with nanobodies. Here we can use the broad portfolio of our abberior dyes, for example the abberior star dyes, which are based on rhodamines and which can be tuned by the addition of ring systems or a larger ring system, for example, or the addition or exchange of groups or heteroatoms in or at the ring system, which leads to a shift of the emission and excitation spectra to the blue or the red region. A combination of dyes that are very suitable for multicolor imaging is, on the one hand, the abberior star 580 dye, which can be excited with the 561 excitation laser, with the abberior star red dye, which can be excited with the 640 nanometer excitation laser. Both dyes can be spectrally separated with these detection windows and used very good in combination with the dead 774 nanometer laser. So here I have an example of a two-color sample where the nuclear pore complex, or a protein inside the nuclear pore complex, was stained with a primary mouse antibody in combination with a secondary nanobody using our abberior star red dye in magenta, and the Golgi was stained with a primary rabbit antibody against a protein in the Golgi or at the Golgi in combination with a secondary nanobody with our abberior star 580 dye. What you can see here is that there is no crosstalk at all and no cross-reaction, and also a very nice improvement in the resolution when we compare the confocal image and the STET image. To add now more than two colors, we are usually limited when using antibodies by the combination possibilities of different species or antibodies of different species that we can use. We overcome this by using secondary nanobodies, or the VHH fragment antibodies from Jackson ImmunoResearch in combination with our dyes, by a so-called premix and stain protocol. Here we can premix our primary antibody together with our secondary nanobodies carrying our dyes, and we pre-assemble a complex of both the antibody and the nanobody before we add this to our sample. Thereby, we can use different primary antibodies, all from the same species, to target different structures in our sample, and thereby circumvent the species limitation and gain a high flexibility in our staining protocol. To add a third color now, we can highly recommend to use the abberior star 460L dye, which is a Longstokes dye and can be excited with a 488 nanometer excitation laser, and the emission spectra then is shifted to the red, meaning that we can use the same detection window as for the star 580 dye. This dye can also be used very good in combination with the, or very nicely in combination with the STED 774 nanometer laser, meaning that for this three-color staining we need the 3 excitation lasers, these 2 detection windows, and only 1 STED laser. So here we can see an example where 3 different structures in the cell were stained by using all mouse primary antibodies. So in the beginning here we have a primary mouse antibody against vimentin in combination with the secondary nanobodies linked to our abberior star red dye. Then tubulin is stained again with the primary antibody from mouse against tubulin in combination with the secondary nanobodies with our star 580 dye. And also the sDNA was targeted here with a primary antibody from mouse together with a secondary nanobody carrying our 460L dye. What you can see here, again, is a very nice improvement in the resolution between the confocal and the stat image, and also that there is no crosstalk and no cross-reaction, showing that the premix and pre-stain protocol works very well and that there is no migration of a secondary nanobody from one primary antibody to another one. So overall, this shows very nicely that the combination of the VHH fragment antibodies from Jackson ImmunoResearch in combination with our abberior dyes offers very good results for multiplexing and for super resolution microscopy. As a last point, I would like to give a short outlook on another possibility to use secondary nanobodies, which is the min-flux microscopy. Here we can reach even higher resolution compared to stat microscopy of around 2 nanometers. And what you can see here is the nuclear pore complex, which is stained by a primary rabbit antibody against a protein in the nuclear pore complex in combination with a secondary nanobody stained with our flux 647 dye, which is very suitable for min-flux microscopy. And this is something that will to combine Jackson VH8 fragment antibodies with our flux dyes to offer new opportunities also for min-flux microscopy. Overall, with the secondary nanobodies in combination with our abberior dyes, we offer now very good results for multiplexing and also for super resolution microscopy. And I will be very happy to answer all your questions in the Q&A session, and thank you for your attention.
So now we've reached the end of the pre-recorded portion of the webinar, and we'll move on to the live Q&A section. So now we can ask some of the questions that have been coming in the chat during the talks to our speakers. We will answer all of the questions. If not live, we'll get back to you by email. And thank you very much for joining us up until now.
Okay, hello everybody. Welcome to the Q&A live portion of the webinar. Thank you for sticking with us up until now. So I've had plenty of brilliant questions coming through, and I'd just like to say thank you again to our two speakers, to Kristina and to Katie, for the great talk that clearly a lot of you are very interested in. So let me see. Okay, so I have a question asking if the brightness of the staining with secondary nanobody dye conjugates is the same as with normal secondary antibodies. So I suppose Kristina probably have more experience with the brightness there.
Mm-hmm. Yeah, thanks. I hope you hear me. So in principle, we obtain with the secondary nanobodies a very good brightness due to the polyclonality, as Katie already mentioned in her talk. And we compared the secondary nanobodies to staining with secondary antibodies. And here we found that depending on the dye and also on the primary antibodies that we use and the structure, that we have a comparable brightness or a brightness that is like 3 to 5 times decreased. However, by increasing laser power or line accumulations, we get excellent results with the secondary nanobodies.
Great, okay. And have some questions about fab fragments. So two that kind of come together. So what's the difference between a VHH and a fab fragment? And are you producing VHHs also specific against fab fragments?
Yeah, I can answer that. Can you hear me okay?
Yeah, good.
Okay, great. So the difference between VHH and fab is a VHH actually is a fab fragment. It's just a fab of a heavy chain only antibody. So traditional fab fragment has both a heavy and light chain, but it's the portion of the antibody that basically you digest off the FC portion, and you have the epitope binding region left. So it's a monovalent. And as a secondary specific VHH, so these VHHs that Jackson is producing right now are heavy and light specific, so they'll recognize epitopes all over both the heavy and light chain. But in the future, we do have some products in development where we're looking at fab specific and NFC specific. So it's not something that we have available right now, but hopefully down the road we would have something like that.
Good to hear. Okay, so I have a question regarding the premix and stain approach. So it's asked, I'd be interested in the recommended ratio of nanobodies to antibodies in the premix and stain approach. And are there any restrictions for using this protocol?
So I think I can answer this. So we recommend a ratio of secondary nanobodies to primary antibodies of 6:1 or to 10:1, so in this ratio. And concerning the second part of the question, so it's not easy to make a concrete predication to this, since the success may depend on the experimental conditions, including, for example, the choice of the required primary antibody that you want to use in your experiment. So in our testings, for example, that I also showed in my talk, with all primary mouse antibodies in this premix and stain protocol against vimentin, tubulin, and the nuclear pore complex, yeah, it worked out very great. So we got great results with this. However, depending on your experiment, it might need or require some individual optimization. For a more detailed protocol and for everything that you want to know regarding handling, you can also check our website. There you will find instructions concerning the premix and stain protocol with nanobodies, but also a protocol for yeah, this conventional sequential way of staining with indirect immunofluorescence, also with nanobodies.
Okay, and another one for you, Katie, I think. How do you deal with the batch-to-batch variation of your pre-absorbed VHHs? And how sure can one be that there's no cross-reactivity in my sample?
That's a good question. So we get that a lot. For polyclonals, we've hit the nail on the head that batch-to-batch can be an issue. So we definitely have this all through our QA area where we test every batch the same way and make sure they're all to the same specifications. So our cross-reactivity testing that we do for every batch before we release that antibody batch has to do with the percent reactivity of the off-target versus the percent reactivity of the on-target. So say you're looking at an anti-mouse and you don't want it to react with rabbit. So mouse would be considered 100% signal, and your off-target rabbit would be less than 1% of that on-target signal. And that testing is done primarily by ELISA. But if we have, sometimes we test in other ways, but every batch is tested that way.
Okay, and a more general question, could we use these nanobodies with any primary antibody?
So that's going to be just basically limited by the species available. So right now, Jackson has VHHs against human, mouse, and rabbit IgGs. So if your primary antibody is a human, mouse, or rabbit IgG, these will work well. And as I said before, they're heavy and light specific. So if it's, say, an SCFV or something like that, where you have just a Fab region, or you're looking at anti-human FC because you have a human FC tag. So these antibodies are both heavy and light specific, so they should recognize epitopes, again, all over heavy and light, and be able to find any of those antibodies as long as they have those host target IgGs present.
Okay, and I have one asking about the Jackson Elixirfluor 790 nanobodies. Do these work well for immunofluorescence too, or are you implying that they only work well on blots and flow cytometry?
Yes, sorry for the confusion. So generally, our customers are using them for flow cytometry and Western blotting, but certainly if you have the microscopy set up, so you have the proper lasers and filters set up, you could use it for immunofluorescence as well. You just have to make sure you're optimized for the fluorophore that you are choosing.
Great. Okay, and I think a final one for Kristina. Do you have any recommendations for blocking solutions that should be used or avoided with nanobodies?
Mm-hmm. So I think also here a general statement is difficult to make. So this also highly depends on your sample and your primary antibodies and so on. For our experiments that we did, we used 3% of BSA, which worked out very well. For special applications, like for example, mouse-on-mouse labeling, also other strategies are required, like blocking with FAB fragments, for example. And yeah, as I already said, a detailed protocol also concerning this blocking strategy you can find on our homepage.
That's great. Okay, so I think that's probably what we have time for in the live question answering. But if your question hasn't been come to you so far, don't worry. As we said, we will be coming to those later on via email so that everyone's questions will get answered. So it just remains for me to say thank you very much again to Kristina and to Katie. And also that the chat box will remain open for another half an hour or so if you have anything that comes to you that you'd like to ask. So feel free to contact us. So thank you very much to everyone for attending.
Thank you.
Thank you. Thank you, everybody. Have a great day.